Well, thank you very much to the organizers for allowing us to share a little bit of what we've been doing in the past decade, trying to foster some of these transdisciplinary approaches with the hopes of trying to get some new science specifically for Cancer Institute. And just to put it into perspective for everybody here, I think when we take a look at that landscape, it's something that affects us all. I think it's very hard for anyone here to imagine not knowing someone, heard of someone that has been impacted by the disease. About 1.6 Americans will hear the words that you have cancer this year. And when we look back against some of our other sister agencies with respect to how we have been doing, this is what really drives us and sort of what made the Institute think about formulating a concept shop back in 2002. And again, coming back to the comment earlier, it's also not something that just affects us here, but globally. I want you to really focus on the purple, where the percentage increase in cancer deaths since 2002 has increased by over 50%, some of which to that 100% mark. And again, as Melissa already mentioned, it is by far the probably most exciting time to be in science, or the science of omics, since its first adaptation or introduction in 1923. We've almost managed to outpublish ourselves just between 2005 and 2010. And for those of us that saw this recently, in November, the publication of 1,000 human genomes being done, there's really a lot of information out there being generated. And sort of follow up a little bit on what Melissa's mentioning, is this really yielding more solutions for the patients? My chart is a little bit less rigorous than the one that Cyril showed, but what I really want everyone to focus here on, and the pharmaceutical partners I think will know this, is this steady decline in new molecular entities that are coming out, and is that a gauge of that innovation? And on the other side, looking at biomarkers, yes it is, but for us, could we make it a little bit more efficient is what really drives us. And of course, those that saw the publication back in 2011, specifically looking at oncology, there's a lot of discussions from our pharmaceutical colleagues saying that a lot of this was inconsistencies that they could not replicate within their labs, juxtaposed with some of the nominal amount of data that could actually fit in line with their in-house data. But I just wanted to remind everyone here, obviously since we fund a lot of our public sector research institutes, that over the past 40 years, 153 of those approvals have been a part in these public sector research institutes, and we feel that that innovation is still there, and we're hoping to figure out, obviously, ways better to take that from the institutes out into and benefit patients. So when we took a look, and obviously we at the Cancer Institute, and we being limited by budget, we really didn't want to try to solve everything, but we did want to try to find ways to intersect perhaps in these early areas at the Cancer Institute. And we reached out back to the community in 2002 to ask what do you think are those key needs that would really take us from one place to another. And obviously what surprised us a little bit was that everybody wanted standards and protocols. At that time, they wanted real-time public release of data, which I think we take a lot for granted nowadays. Large multidisciplinary teams. They wanted a pilot-friendly team environment where you could share failures and successes, which I think, coming back to the research park thinking or the Bell Labs, the ability to sort of share what didn't work quickly versus waiting for that publication to come out was something that they asked us. And last but not least, they wanted us to build teams that the team members themselves were transdisciplinary. Obviously, these were 5 very large bullet points then, but they told us if you could accomplish just a piece of this, I think there was a chance to potentially transform how we do cancer drug discovery and diagnostics. For those of you guys that do not know, we are part of 27 different institutes. Some of our sister agencies are here. The National Cancer Program is not just us, clearly. We consist about a quarter of that, with a piece of it being private industry, and we heard a little bit about the 2% coming from the foundations, which I find for us definitely help us think about that landscape. Specifically, the Cancer Institute itself, what I represent here within the Office of Director, we're a very small organization, 4%, and we have always been driven by the fact that everything that we do has to benefit all of our other divisions, within the institute, and we have the spirit of always spinning successful concepts once they get to a certain stage out into that area so that it could be carried out not just at one level but at all sectors of research. And I think we're doing that actively. Right now I have sort of a split personality in which I still manage the advanced technology sectors and some of the programs and program directors for those, as well as our newly spun out Office of Cancer Genomics, or Center for Cancer Genomics, through some of our initiatives that we launched back then. And all of the lines here, one of the things that I think separate us from some of our other agencies or other NIH counterparts is that all of our programs are asked to be renewed and they have to end. We have to have them sunset. And each one of the red dates here are the times when our programs go back up for renewal and go back up to make sure that they're still relevant, that they're still at the cutting edge. And if they're not, we do stop them. And this is where I think when I first talked to Cyril about the conference itself, I asked her, you know, which ones do you really want us to talk about? I won't get a chance to go through all of them, but I think for the sake of what we're talking about here, we have programs that obviously help support discoverers and pioneers. I won't get a chance to get into those, but I will focus mainly on these three initiatives that really are those first team explorers out into the Wild West that haven't yet settled like some of our more advanced initiatives, like our nanotechnology initiative that is really cranking out some first-in-human trials of nanotechnology platforms for cancer. So how did we actually take that very first step? I think we did a little bit of a gauge or a dashboard back in 2003 with the launch of our innovative molecular analysis technology program. And there, a lot of them, when we first launched this program, not too many of the technologists believed that we really wanted proof of concept technologies that had minimal biology, but really said if this worked, it would have some impact in this emerging arena. And we ended up looking through what came in the door. I'm not going to say everything was exciting. There was still about a quarter that was sort of the same old, same old. But we saw a lot of genomics then, both in the innovative category as well as the emerging category. Some proteomics platforms that were starting to emerge, nanotechnology platforms, and some physics platforms. And we pretty much just went 1, 2, 3, 4 and launched successive interdisciplinary programs in those particular arenas. So that very first one is almost a first step back, taking a look at what everybody thought about cancer genomics at that time in 2002. Obviously everybody had their own way to try to assess what was changing and how heterogeneous and complex the disease was. So we thought, well, let's take a page perhaps from engineers and make perhaps a reference table. Let's do a systematic identification of all of these, or as many genomic changes as possible. Repeat it a lot for each individual cancer, repeat it for many cancers, and make that data real-time publicly available. You can imagine I can stand here and say that this was easy now, but back then we had articles written about how much of a waste of the time and money the Cancer Genome Atlas would be. And what we did there is we took 3 pilot cancers, GBM, lung cancer, ovarian, and asked for multiple data types, asked for the same piece of tissue to be sent out to each of these characterization centers, and then have that data pulled back in. And the very first paper was published in 2008, and I would say one of our small innovations was trying to have them all published under one name. So everybody took credit together for this first reference cancer genome for GBM. Some of you guys have already seen some of the outputs of people using that reference genome, both in finding exclude non-responders, potentially better prediction of outcomes, but not using the data set alone, using it as a reference against your own data sets. So really having people involving their own data, not just the data that we put out there. For myself, I'm a chemical engineer, as I mentioned, so this is just my way of showing to everyone here what our progress is to date. So for those that are not familiar looking at this type of data, I do want you to not look at the details. I want you to look at the patterns. So each number here is, and each row is one patient. So the numbers here represent the amount of patients within each row. And you could tell we really got lucky by picking GBM as our pilot to be able to demonstrate that this worked. Our second one, ovarian, is a little bit more heterogeneous. This was summer of last year, and this is 2,000 samples. This is spring of 2012. We're at about 4,000 samples. And this I compiled myself, so it pretty much demonstrates to you guys that anybody can go on and download this data set. You don't really need much specialized knowledge to be able to get that first look at the data. And where we are, we had a great meeting last week. Dr. Kenneth Shaw continues to help lead this program for us. This is where we're at with respect to what's up and coming. We're hoping to finish all 25 tumor types by 2014, and the program will end. But I'm hoping for you guys that have interest in this data space to begin perhaps taking a look at what's up and coming in this arena. I just wanted to share with you guys to close out this example. Last week at this science symposium, it's very interesting to see that STEAM table, or that cross-cutting way of looking at the disease from two different perspectives, from the academic perspective that was presented, as well as the industry perspective that was presented. And I think if you take a look, both of them arrived at a similar conclusion, that some of the mutations that were not quite evident, not the big hits within a disease, ended up being very relevant if you looked across the different diseases. They kept coming up, but they weren't the ones that came up at that very hot spot zone. And I think the RAF example given by our industry colleagues, so that they weren't able to sort of tease this one out until they actually started stacking these genomes against each other to try to look for those differences. So what about biomarkers? You know, I think we talked a little bit about what we did trying to advance perhaps target discovery. This one was more of a step 1.5. National Academies, I think, put out back in 2002. For those of us that have owned and or used a mass spec around that time, this won't come as a surprise, but at that time, the proteomic technologies were not quite yet mature to do what we would like to have done in TCGA. And nothing tells you that better than having a whole bunch of great labs back in 2005 demonstrating just how many things they could sort of verify with one another. Unfortunately, it wasn't very much as you kept going up. And what we really wanted to try to get through there with a program, I won't get a chance to talk to you about the phase 1, but the phase 2 of the program, after going through 5 years, working with the FDA and working with several investigators, key leaders in the mass spectrometry realm, they were able to finalize a way to be able to share data and to consistently get reproducible data. And what's exciting from my perspective, and this phase 2 was launched in September of 2011, those same TCGA samples that I showed you before now will be concurrently analyzed with proteomic platforms. And as you can imagine, for those of us that have dealt with proteins versus genomic materials, it took some time to make sure that how we collected tissues for genomic analysis, you could use that same piece of tissue that was fresh frozen, that all of the phosphorylations, the proteins, et cetera, actually stayed intact. And that's what the group spent a little bit of 2011 doing, being able to say, I took that piece of tissue that was frozen. Could I see things that the genome group had seen already? And I just wanted to show you a little bit. They did their usual data dump, and when they convened and discussed and looked at the data, obviously these are all the groups that submitted their data. And I think what's exciting from my end is once they started analyzing the data together, some of the things, again, that looked like noise became real signal. And obviously, if you can't see a theme already, the public data portal is open now with the phase 2 data also uploaded there. These are raw data sets, not already analyzed. Henry Rodriguez helps me really spearhead that program, and a lot of this type of thinking, I think. I've told him and his group that if he doesn't find things that the genomics group didn't find, then it was a waste of time. So obviously they are not just trying to verify what TCGA has found, they want to go and find things that TCGA didn't find. So I think some of you guys may have participated last week. Our director, Dr. Harold Varmus, held a meeting to try to ask this question. I think everybody's asking What's next? Where do we go from here? We gave you genomics data, we gave you proteomics data. Is it now we begin, and these are obviously fake data, is it to do now time evolution data? Is it to delve in single cell and really get into that intricacy? I think from where we stand, since this is a problem that I think everybody is working on, what I wanted to share with you is I personally think it's the people. It's not the next scientific project. So the program I wanted to share with you actually, and the reason I'm showing this, not just to show you why we continue to think innovatively based on response from the outside, but the day that Gina published this particular article was the day that we reviewed, I believe, one of our riskiest programs that we've ever launched at the Cancer Institute. You heard a little bit about it from Nicole. Back in 2009, we launched an initiative with the charge of generating new knowledge and catalyzing new fields of study by bringing in physical scientists and having them lead the team and work with physician scientists, clinicians, and bring how they think the disease works and prove it. And I think Larry Nagahara had a chance to present at FastaCure, so I had a chance to showcase a little bit some of the things that we're doing. But I wanted to show you each one of these physical scientists to your left, or yes, to your left, is paired with a physician scientist, a clinical oncologist, that really helps them guide through their framework of thinking. And I would say just giving them that partnership was insufficient. What we ended up doing was crafting a couple of mechanisms that NIH at that time was not comfortable with us giving to PIs, which was giving PIs the ownership of how they gave out money, in the sense of having them innovate with us. What I mean by that mechanistically is basically a bunch of set-asides in red that we asked each of the applicants to have. They could write no science against that. They could not put any specific aims to that money. They had to put that money aside for specific purposes. That was very uncomfortable for NIH in the sense that it was not peer-reviewed because there was nothing to peer review. But we were able to get that mechanism out and test it for this particular network. And a couple of the mechanisms include exchanging trainees. They had to do that. They could not use it for any other purpose. They had to design per year their own pilot projects. And we did not particularly care what they spent that pilot project money on, only that they spent it fairly. They did not give it to their friends or people that they just knew. Some of them have used it to kickstart potential pilot ideas within their centers. Some of them have used them to replace actual big projects. I think one of the places that we have found most exciting is that we asked all of them to ante $100K to the table for a full pot of 1.2 throughout the 12 centers. And they were only able to access that money after designing how a transnetwork project would look. And the people that did not want to participate, and they said, look, this is too much, I'll ante up, but I won't use that money, that has happened. So some centers have had to give their money to someone else. I think a big skepticism that we get a lot is nobody would ever do that. People have. I think another interesting part are these outreach pilot projects that we've given them. So when they go to a conference, when they go to a meeting, if they meet somebody that they find particularly exciting, they can bring them into their network immediately. And that, I think, in our opinion, really begins accelerating some of that thinking and sharing of thinking out into the community that perhaps were not funded as one of these 12. We also did not lose our minds. So as you have heard before, all of our programs need to be evaluated. So we worked with our own portfolio analysis office, folks inside here, to develop a unique prospective and simultaneous evaluation that, again, NIH had not seen before, in which we watch, track the program and give feedback to the investigators through reports that Tom is here, that I know are long and arduous. But at the time, we got the investigators buy-in. We told them that we wanted to do this so that 5 years down the road, how many of you guys remember that the iPhone was not particularly well received? Nobody remembers that anymore. So I think we wanted to make sure that when everybody thought that physics was important or not relevant back in 2009, everybody thinks that way now, we are able to sort of track that. And Nicole is here if you have questions on this particular tool that she has built to be able to not only look at it, but be able to give back to the investigator how they're doing on a 6-month to 6-month basis. I just want to give you a snapshot of what that looks like. So some of our networks, and this is a static, the tool itself allows you to really play around. Each of these things can be then tracked back to an actual publication. But the evolution of these centers over time, I think is one of those things that we're asking evaluators, this community perhaps, to help give us an answer on what's good. I mean, it's very hard for us to say, is this much collaboration good if we don't have something to metric against? And other static type images that we give back to our investigator are sort of the evolution of trainees over time. What are they doing? Where are they going? And I'll show you something a little bit perhaps more interactive. This is one of the things that we're seeing coming about through this network. All 12 are represented here, and each single number gives you a sense of who's leaving and where they're going. And this was summer of 2010, winter of 2010, summer of 2011. And in my mind, this transition, spread, thinking, training of these physical science and oncologists that we tend to call them now, I anecdotally call them hybrids, I think are very exciting to see where they're going, where they're making that impact. And perhaps back then, when the Human Genome Project was decoded, I think there were 4 guys, I don't want to use any age references here, but 4 guys staring at the 3 billion pieces trying to figure this out. I just wanted to show you, now in 2012, I took a look back at those 3 papers of TCGA and watch how that has morphed. And every single paper from that pilot program, and I'm pretty sure the next one that comes out, we're going to now start hitting that Moore's Law of Analysts, being able to find the right people that have both sides of the training to be able to mine that data set, not just within the disease, but across the disease, is something we're hoping both of these programs can be synergistically thinking about, and perhaps this group can help us think about as well. So just to close out, I think, as you can see, I've shown you guys, I think, a couple of projects that when we first launched them, the creativity over status quo was pretty high. It was very easy for us to sell. I think programs like TCGA, once they got to a point of, hey, this project works, the creativity is not that great anymore, it's a little bit harder for me to say, am I more excited about my physics program or am I more excited about my genomics program? Because in my mind, I never get a chance to hear where they're particularly at. I think we do a lot of juxtaposition without saying, what did it look like then, and can you compare them at the same time points? Another way perhaps to say it is that we tell our investigators here to go through this funnel every single time. And those teeth can represent anything here, FDA, peer review, your choice, being able to cross that valley of death, and hopefully when they come out on the other side, they're willing to do it. Unfortunately, we keep asking them to do it over and over and over and over again. Each time we take a basic scientist and bring them through this space into perhaps the translational realm, they again have to learn new things. They again find new jaws and teeth that they have to hit. And one of the places that I think why we are as successful as we have been is that we don't treat these folks down here that didn't make that leap as folks that didn't make it. We treat them as now teachers who can now inform the next set of folks that are coming through. Maybe they went through it and they said, you know what, this is not for me. But they, I think, have an extra set of insights that they can give to the next crowd coming in, saying that's perhaps what it looks like. And I'm hoping that is why, coming back to my chart before of where, when we launch some of these programs, we expect them to sort of be, versus a few years down the road where they end up being. One of the things that I'm most excited about is the fact that in our physics program, we are now seeing what I like to call a hopscotch, in which the physical scientists are providing such insight to the clinical oncologists that they don't go through the traditional translation route anymore. They are now designing their trials a little bit differently. They're using different ways of trying to find perhaps unorthodox drugs to include in their trials. So how do you begin looking at this sort of hopscotch phenomenon in which pairing these two disparate insights are coming up with things that don't fit into that turn the crank kind of thinking. Last night I had a chance to go with one of our physical scientists to a hospital, and explained to me last week he spent learning how to do bone biopsies, and it was just something very fascinating for him because in his mind, a bone biopsy was just something that his pathologist would get him. Coming back to some of the worries that I have, some of the things that I think are coming about in terms of metricing and evaluation, and why I find the tool set that Nicole has built so important, is that we tend to immediately say how well a program is doing by looking at their publications. We're almost tenure tracking our own programs. A program like TCGA that may have 1, 10, 5 per year, is that any more or less better than our physics program or nano program that cranks these out into that 80 or 90,000 pool that I mentioned before? And of course, this goes without saying, a lot of the time, timing is everything. When I first made this chart for our new director, I showed him what it looked like. This dotted line here would be what NIH Tech Dev would be like without us. And I was almost hesitant to show this because it made it look like we jumped in when everybody jumped out. And I think from his mind, he thinks it's great because if we weren't going to do it, who else would have? So speaking of resources, we have ongoing programs I didn't have a chance to touch upon, but this year we have another $40.5 million on the table for folks to think, to be in that early discovery phase. For those of you guys that are seeking or looking or know people that need funding resources to do something high risk. And the secret ingredient in my mind is not so much the processes as the people that we have involved over the years. The clinical scientists, the physical scientists, and life scientists that run our programs on a day-to-day basis. We don't have just one type, and I really want to give thanks to my former mentor, Anna Barker. Some of you guys may know her in some of her vision in trying to put all of this together. Thank you for your time, for listening, and hopefully I can answer some of your questions. If you'd like to learn more, please visit us at the website, email me, and I'll take some questions.